So what I want to do now is just, I think probably a little bit more mathematical, I hope that's okay, and talk about spatially resolved models of tumour growth. Again, trying to keep things sort of fairly simple. And as before, well, you seem to be quite comfortable asking questions, so that's quite good. I'd rather do less and answer your questions than you not understand if you want to understand. So tumour spheroids are, I guess we all know, a sort of centerpiece of many experimental labs, routinely used to characterize tumour cell lines, test drugs, learn more about biology, and a lot of that down to the fact that they grow reproducibly. I guess typical dogma would be we supply nutrients in the culture media surrounding our clusters of tumour cells, nutrient diffuses in, provides the cells with, enables them to proliferate, so the spheroid gets bigger. As the spheroid gets bigger, the amount of nutrient reaching the center decreases, so the cells there have less energy to proliferate, eventually insufficient to remain alive, and so they will become dead or necrotic. And so with a fully developed spheroid, that would be characterized by, or might be characterized by, an outer shell of rapidly proliferating cells, which are increasing the tumor volume at a rate which balances the rate at which dead cells in the center are dying, okay? So again, the growth and structure is reproducible. We saw this in the previous talk. So typically initial exponential growth when all cells are nutrient rich, plateauing off when we reach a balance between the rates of net rates of cell proliferation and cell death. Yes. Yeah. Do nutrients include? Yeah, whatever. I mean, I guess I'm focusing here on perhaps a growth rate limiting externally supplied nutrients. So that might be oxygen or glucose or something like this. But yes, there could be other things. For simplicity here, we just focus on, like, I will typically talk about oxygen, but I guess it could be other things as well. If you're saying that there are other byproducts that are produced locally that could sort of also fuel cell proliferation. So for example, I guess when cells die and degrade, then they will produce materials that can be taken up by other cells to provide the biomass for cell growth and proliferation. But I guess for simplicity, I'm really at the moment thinking about a single growth rate limiting factor, which I'm calling oxygen, right? So really what I'm doing here is we're going to start with, I think, some of the earliest classical models of spatially resolved models of tumour growth. And this is going back, if you can think back that far, to the 70s, so quite a long time ago, where our knowledge of the biology was quite crude. So I guess that's where these initial models come from that I'm just going to try and introduce. So here's a time course of spheroids showing pretty much what I've already described. If we want to try and build models to describe these processes, again, so as I said, the model that will sort of, one of the starting points for this section is a model by Greenspan from the 70s. So yeah, that's like 50 odd years ago now. And at that stage, it was very much about, I will stick an oxygen probe into a tumour spheroid. I can just about measure the oxygen tension and maybe lactate, but it was very, very crude and very, very limited data. And so I'm going to sort of introduce in a general form the sorts of models that were developed with based on, I guess, the hypotheses that we've got here as to what's rate limiting. But again, as with all of biology, this is just very, very simple, but we all make approximations when we're building models. The art is what's the minimum number of assumptions or processes to build into my model to make it physically realistic and to answer the questions that I'm trying to answer, right? There's always more biology that we're ignoring. So that, all of that aside, so here, simple models from 70s. So the assumptions are that there is a single generic growth rate limiting nutrient which is supplied externally. It diffuses through my cluster of cells. As it does so, it will drive cell proliferation where it's high. It will drive cell death where it's low. As a result of all of that happening across the tumour volume as a whole, the tumour will change, increase or change in volume. And so therefore, if you think about it, what we're doing is solving, say, a reaction diffusion equation for this diffusible species nutrient, oxygen, but the domain on which we're solving that problem, the tumour, is growing, it's changing. So it's not like a classical reaction diffusion equation on a fixed domain. We're solving it on a domain whose size changes, and we call that a free or moving boundary problem, right? So as part of the problem, we don't just determine what the nutrient concentration is, we must look at what the proliferation rates are, what the net growth rates are at different positions through the tumour, and that integrated will tell us whether the tumour is going to increase or decrease in size or reach an equilibrium. So it's a moving boundary problem, which is a class of problems that are beloved of many applied mathematicians. So a lot of work, analysis done around these classes of problems. So given that description, what sort of model? So again, I'm just trying to keep things short. I could spend a whole load of time talking to you about the solution of these problems, but I kind of want to do a little bit more than that. So I hope you will indulge me. And if you want to know more, I'm happy to chat later. So given the sort of verbal description that I've just given to you, what sort of model might we write down? So C is my single growth rate limiting nutrient. It's diffusing, it's being consumed by the cells. And I've made an assumption here, a quasi-steady assumption, so I've neglected the d by dt in here by suitable non-dimensionalization. What does that mean? It means that I've scaled lengths and time scales in such a way that the time scale on which the nutrients are diffusing and or being consumed is much faster than my time scale of interest. My time scale of interest is the time scale on which I see changes in the tumour volume, right? And again, this is in the literature. If you want to go through and see the details, but I hope you understand the intuition. And so we can set this d by dt equals to 0 here. Effectively, what we're saying is that the nutrient adapts to changes in the domain size rapidly, so it's as though it's reached an equilibrium, right? So given a particular domain size or tumour size, say radius capital R, I can work out what the nutrient distribution is within that particular tumour volume, and then I update my tumour radius. It responds, and it responds according to this funny looking integral differential equation. So it looks weird, but let me decode it for you. So at a given spatial position, X, within my domain, which lies from X equals 0 tumour centre to X equals R, outer boundary, I can read off what the local oxygen concentration is, the local value of C. If C is high, then I produce more cells, the volume will increase. If C is low, then cells will die and the volume will decrease. So all encoded in this Sn of C, this function, this is giving you at a given location with a particular value of C, it tells you whether or not you've got net growth, so nutrients are high, or whether you've got cell death, nutrient levels are low. Okay, does that make sense? So this will range from negative values when C is low to positive values, increase in volume, C is high. So to get the total growth rate, I just integrate across the whole of the tumour mass, and that gives me the net growth rate. Does that make sense? Okay, so we have a diffusion equation, which is defined on the domain from x equals 0 to x equals r. R is a function of time. R evolves according to this integral differential equation. So it's a slightly weird, non-standard type of PDE, and this is part of the reason why a lot of analysts have been interested in trying to sort of analyze existence, uniqueness, etc., properties of these types of equations. Again, if you're interested, happy to point you to the literature. So these models and their extensions have been widely studied, but this is one canonical, classical type of model that you might use. Again, I haven't really told you anything as yet about specific functional forms for this net growth rate or the consumption rate. Again, if you go through the literature, we can assume very simple functional forms for these terms, and we can make a lot of analytical progress. But I guess in the interest of time, I'm not going to go through that here, because then it feels very much like you're back in an undergraduate or something. So these sorts of models can be fit to the data. Here's an example. You can introduce different sorts of structures. You can, if you like, so this is the outer tumour radius, capital R. You can distinguish, if you like, so this second dotted line is telling you where the interface between proliferating cells here and necrotic core, like this central region here. So you can develop multi-layered, multi-structured models that generalize this. These internal boundaries are defined or can be defined as contours of nutrient concentration. So this curve here, the necrotic interface, might denote the concentration at which we switch from net proliferation to net cell death. Okay? So that's a basic structure of the model. Really, it's a diffusion equation coupled to an integral differential equation. Right? That's if you want to characterize it. Now, if you went and talked to somebody else who does PDEs and said, here's how a tumour spheroid grows, and please will you model it for me? So maybe one colleague would have given you, Greenspan would have given you this model, but somebody else might have given you a different model. And what might that other model have looked like? It might have looked like just a nonlinear diffusion equation, right? So rather than this integral differential equation defining capital R of T, here what I've got instead is N is my cell density at a given position within my tumour domain, and I have nonlinear diffusion, and I have a net growth rate, local growth rate, which depends on the local cell density and the local nutrient concentration. And I couple this PDE to my nutrient equation. Again, quasi-steady here, because the timescale for nutrient diffusion and consumption is much more rapid than the timescale for cell proliferation. Okay? And again, you could think of a constant value for the diffusion coefficient or the cell motility, or you might think of some nonlinear motion term. Okay? So the point being, there's more than one way that you might approach modeling your tumour spheroid. And if you do that again, you can, with a nonlinear diffusion coefficient like this, you can also cast this as a moving boundary value problem. So I guess there's a question then, those models look quite different. Is there any sense in which I can sort of unify them? Any sort of theoretical foundation that kind of is common to both of them? Can we establish, and furthermore, if you think about what we were talking about before the break, these models are treating my tumour as homogeneous. There's only one cell type, only one species. Tumours are not homogeneous. I'd really like a framework that I can generalise, and the different species within the tumour, they may have quite different properties. I could just write down a whole load of reaction diffusion equations, and indeed that is perfectly fine, but I may also want to take account of mechanical effects. How can I do that, right? So cells respond to pressure. If you have blood vessels and you have pressure being exerted on them, they may collapse, they may close off and not supply oxygen anymore. How could I do that with these sorts of models, right? So I want a more general framework that might allow me to study those sorts of systems. And so that's what we're going to talk about for the next 40 minutes. And so this is really all around multi-phase modelling, which is an idea that's been used very much in industrial applied maths, applied to soil growth and lots of different applications. And really what we've done here is specialise it for biological systems, particularly tumour growth. There are many other biological applications in terms of tissue engineering, all sorts of things. And in the first bit, I'll just introduce the basic idea and then show you how that sort of approach might give rise to the Greenspan and the reaction diffusion model, and then we'll look at extensions. Okay, so sorry, this is going to be a little bit mathematical. I've tried to make a gradual reveal. I thought about doing it on the board, but I thought that would be too horrible. Or too much writing and you might not be able to read it. So the idea here is think of your tumour spheroid as a mixture of cells and water, okay? And I'm going to write down mass balance equations for both of those species. So, oops, how did that happen? So mass balances, I'm assuming that the cells move with a velocity Vn, and that the net rate of production is some function Sn. The water, the mass balance is the same. Notice that I'm assuming here that the only two materials are cells and water. So if the cells are growing, that's done by taking up water. So there's an exchange of phase between cells and water. When a cell dies, the dead material is water. Again, gross assumptions, but mathematical approximation, indulge me. So these are my PDEs that tell me how N and W evolve, but I need equations for Vn and Vw. So we can write down momentum balance equations. Sigma N and sigma W are stress tensors. This is a 1D model, so they're just scalar quantities here. So those describe the material properties of my cells and my water. We'll come back to that in a moment. So we have our momentum balance, neglecting inertial terms, is we've got internal forces which are captured by the stress tensors. We have a drag term due to relative motion between the water and the cells. So if they move at different speeds, if W is moving faster than N, then it will donate momentum to the cells and vice versa, equal and opposite terms here. And then we have this extra term, which is due to interfacial forces. So P is a pressure, and this comes from doing homogenization, volume averaging. Don't want to go into the details here. Again, I'm happy to point you to the literature, okay? So this is what we get by taking a continuum limit, going from, say, cells as being discrete objects to viewing it as a continuum, okay? So I can view the mass balance equations as telling me what N and W are, momentum balance equations for VW and Vn, but notice I've slipped this extra P in, so I need an extra equation to specify that, and we get that by specifying no voids, i.e. space is occupied exclusively by cells and water. The mixture can change, the balance can change, but there's no holes, everything is one or the other, right? So in order to close this model, then it remains to specify the net proliferation term and my stress tensors, sigma N and sigma W, right? Is that okay? Okay, good. So then I guess the question is, how should I view my cells and fluid? And again, probably the simplest thing to do is to assume that both are isotropic fluids. So therefore, I associate with each of them a pressure, Pn for the cells, Pw for the water, right? And I'm going to make the further constitutive assumption that the water pressure is the same as this interfacial pressure that I introduced on the previous slide, That's going to be encoded in SN. So I haven't told you what SN is yet, but we'll get there. And there will also be an equation for nutrients, which will diffuse as we had in the other two models. So really, I'm focusing on the what's happening to the cells here. Okay, so I'm going to, as I said, assume that the cells in the fluid are isotropic and therefore associate with them a pressure, right? Yes, the cells and the water. The cells are moving. They're moving in some way, right? So at the moment, I just say they're moving with a velocity. So again, I haven't said more about that yet, but it will come out.
It's all in one dimension. So
yes.
You're forgetting about any,
you can cast this in higher dimensions if you want. I'm doing it in 1D so that it's simple and you can see the principles. You can extend to higher dimensions. It just becomes a little bit, I think this is the most intuitive way to see the principles. So let's think about how we might specify PN. And I'm going to propose two different assumptions. I'm going to consider two different cases. In the first case, I'm going to say my cells are just incompressible bags of water, or they're just totally incompressible, and they have a fixed constant volume fraction, right? So N equals N star is constant everywhere. Now, if I make that assumption with no voids, then I'm also making the assumption that W is constant, because W is 1 minus N, right? So I'm making a fairly strong assumption here that any place within my tumour, that the density of cells and the density of water are fixed, right? So you might think that's a terrible assumption, but just, indulge me. So that might be one choice you could make. Another choice you could make is, do you know what? I think cells are quite like water. And so I'm going to make an assumption that cells are effectively bags of water plus a bit. And the plus a bit is what makes a cell not just a bag of water, and that's captured by this function sigma of n. And this kind of says something about the way cells interact with each other, right? So apologies for the change of notation. Phi here is really n. And so the idea here is that if the volume fraction of cells is very low, so the cells are very sparse, then I don't know there's any cells around, and that term is switched off. If the cells are very densely crowded together, they don't like that, and they want to repel each other. And so, and then there's an attraction radius here, and so this is the sort of functional form that you might prescribe for this function capital sigma. Okay? So what we're going to do is walk through the consequences of these two assumptions, two different alternative assumptions, and see where we get. That's the idea.
One more question.
Yep.
So then the velocity that Vn and Vn, for example,
yeah.
So then Vn is a function of x, and so it's implicitly a function of n as well.
Yes. Yes. So we need to work all of that out. And so we're going to follow through the consequences of each of these two assumptions and see where it gets us. Okay? So we've got if we go back to the previous slide, so see what I've got down here. I've got two conservation laws, right? So let me just go back. Oops, here. If you take the two mass balances, add them together, you get d by dt of n plus w. N plus w equals 1, so that goes. And so and the Sn and the minus Sn cancel. And so I get d by dx of nvn plus wvw equals 0. Integrate up. Symmetry, no velocity at the center. And so that's equal to 0. I can add my momentum balances together, and I also get a sort of total momentum balance for the system as a whole. So that's what was at the bottom of this slide here. So conservation of total mass and momentum supplies these two identities. Okay? So I'm going to use those. Yep. So all of this will be playing out on a growing domain. Growing domain. And I guess the assumption you would make is that the position of the outer tumour boundary, because the tumour is defined where the cells are, so the domain grows at the velocity on the boundary. So if you like, dr by dt is equal to Vn evaluated at R. Does that make sense? Yes. Yes? So, but again, I guess I want the exponential growth to be an emergent property rather than hardwiring it in. Does that make sense? So, because it's exponential initially, but it isn't exponential forever. It will be exponential, linear, and then asymptote to 0. So I want a model that will do all of that for me because I'm greedy, right? Are we happy? Great. So let's walk through the consequences of assuming n equals n star. So if you look at the mass balance equation, if you put n equals to n star, so sorry, I've just summarized the equations here for you. If n equals n star, then the mass balance equation reduces to dvn by dx equals Sn divided by n star, right? Which I've called Sn bar down here. Does that make sense? So n equals n star in here, I get this equation. So this tells me if I assume that the cells are incompressible, this is how my velocity goes, right? So local changes in velocity are driven by the net growth rate. So Sn, if you remember, is a function of C. I just haven't written that down yet. Okay. Now, if we work a little bit harder, so I've also got my momentum balance, and if you substitute sigma n equals minus Pn, exploit this and do some algebra, then you can arrive at this expression for the cell velocity as well, right? For the cell velocity in terms of Pn, right? Combine these two equations and you get a Laplace equation for the pressure, okay? Now, as we've said, we assume that our outer boundary, x equals capital R of t, is moving with the cell velocity on the boundary. If you look at this equation here, integrate from x equals 0, where Vn is 0, through to x equals R, you'll get exactly this. Right? And what does that look like? Yeah. And so this will be coupled to an equation for the nutrient. So this is exactly Greenspan's model. Right? So what this says is another way to think about Greenspan's model, or what was implicit in Greenspan's model, was the fact that the cell density was a constant. Okay? So, and again, you can solve these equations, la-di-da-di-da. I guess you could legitimately ask the question, you didn't really need this momentum balance because you've just walked down this side, right? And that would be a legitimate statement to make. Does anyone have any thoughts about why it might be useful sometimes to have this equation? Say again? But the velocity, so there's a bit of an inconsistency potentially here at the moment. The velocity is determined by this. Sn is a function, well, so at the moment, so you're correct. So at the moment, or up until now, we've assumed that Sn is solely a function of C. So if it wasn't just limited by nutrients, if the cells were also mechanosensitive, so if they responded to the local pressure, then it would be important. So what this model does is it allows you to consider a wider class of stimuli that might be affecting the cell's behavior. So exactly right, Brill, good stuff. Oh, what does that say? Yes, well done, good spot. Yes, 100% right. So now let's go on with the other. Yes, go for it. So yes, so SN is, if you like, a function of C. So I've kind of been a bit sort of elusive, non-committal about it. But yes, if you want to recover Greenspan's model, it would just be a function of C, and I will couple this integral differential equation with a diffusion equation for C. But we get the extra sort of mileage we get from this framework is it allows us to look at situations in which N might be a function of P, the pressure as well. And it could be Pn, it could be Pn minus Pw. What's driving? So it allows you to explore a sort of wider sort of range of possibilities. Okay. So now let's look at the other case, and no prizes for guessing where that one's going to be heading. So the other closure that we talked about was to say that the cells are bags of water plus a bit, the plus a bit being captured by this sigma of n, density-dependent function. So if you walk through the algebra, so the momentum balance for the fluid, effectively, if you unwrap it, gives you this expression. If you combine that with our mass conservation, which relates Vn and Vw, then you can arrive at Vw is minus 1 over k dp dx, which is exactly Darcy's law. Right? So we're saying this is Darcy flow. If you work a little bit harder, then you can arrive at this expression for the cell velocity. We know that, so again, it's just algebra, so dp by dx, and if you go back and use the cell momentum balance, then you can work out Pn in terms of this capital sigma n. So remember that Pn is P plus sigma n. If you use the momentum balance for the cell phase, again, sparing you the algebra, you can arrive at this expression for Vn in terms of n, okay? So effectively what we've done is we've been able to eliminate P, W we know is 1 minus n, and we can eliminate VW from here, and we arrive at an expression for Vn in terms of n alone. And I guess just to make that a bit clearer for you, here, the total momentum balance in this case, which is sigma double n, W sigma W plus n sigma n equals 0, substituting gives you dp dx in terms of minus d by dx of n sigma n, which is where this has come from. So now finally, if we substitute for Vn in our mass balances for the cells, so so Vn or nVn is equal to this, then we arrive at a nonlinear diffusion equation for n, right? Where Sn is a function of C and whatever else you like it to be. But the important thing being, under this assumption where I treat the cells as a fluid with some sort of sensing of cell density, then I can arrive at this nonlinear diffusion equation for my cell density. And I guess the point being also, additional point here, is you can sort of get an interpretation, you can relate the nonlinear diffusion equation, the coefficient that comes out of this, to your assumptions about the behavior of the cells. Does that make sense? Okay, so I guess the point here was with a simple-ish two-phase mixture model that provides a more general framework on which we can sort of within which we can sit these two sort of what might seem quite different mathematical models, and you can see how the difference between them is very much, or can be thought as being how I describe the properties of my cells. In one case, I treat them as incompressible or fluid-like with plus a bit, and the other case, I assume that there's a fixed density of cells throughout my tumour. And each of those different modelling assumptions gives rise to quite different continuum models at the tissue scale. Okay, and so there's been a lot of work. That's that's really the basics of how these multi-phase models go. We can add extra phases. The algebra just gets a little bit more involved. We can extend to higher dimensions, and indeed all of this has been done, but it allows us to make more general assumptions about the material properties of the different constituents within my tissue. In the models, the two models that we've just looked at, we assumed that the phases were isotropic. We can also think of more general cases where they're not isotropic. We can look, so the reference down the bottom here is where we try and think of the tissues as being elastic, a morphoelastic framework. So there's lots and lots of interesting things that you can do with these sorts of models. And I guess the key thing is it allows you to start to investigate the additional impact of mechanical and stimulus on the cells. How does that affect tissue growth? And additionally, it allows us to introduce additional species. So what I'd like to do now is I'm going back to this data. So you've already seen this morning. And if I now tell you the truth about how that 2D model, that two-compartment model came about, we didn't just go there straight away, right? We had a bit of a journey before we got there. And I'm going to tell you about that journey, right? So what we did was we were trying to understand this data, and what we thought about was writing down a three-phase model. So very similar to the ideas this morning. So we've got cells and water and dead material, right? And so I'm going to write a three-phase model, and then we'll see what happens and what qualitative behaviors does it exhibit, and can it reproduce these data? And that's what we did, first of all. And then we thought, my God, that's really complicated. Can we make it simpler? And then we wrote down the compartmental model, right? So that's how it actually happened. But I thought you probably didn't want that first thing this morning. So, again, this is really just motivating what I've just said. So we're going to view our tumour now as a mixture of live cells, dead cells, and fluid. I can apply mass and momentum balances to each of those phases. And then there's a question of what, so I'll assume that my fluid is an isotropic fluid. I'm actually going to assume now that my live cells are a viscous fluid. And I'm going to assume that my dead material is somewhere between an isotropic fluid and a live cell, right? I'm going to hedge. I'm not quite sure how to characterize it. And part of this will be looking at what the consequences of what the properties of those dead cells are, how that affects the tumour's growth dynamics. Yes? I think, so why didn't we just do it isotropic? I think that, I think we just thought it was a bit more realistic. No very strong reasons. I can't remember what happens if we get rid of the viscous terms. I think it just gives you a little bit more resistance to movement. Mm-hmm. Mm-hmm. Yes, so that's the viscous terms. Yes, yes. I think we were just trying to make the cells be a little bit more than just water. So I guess you could think of it as perhaps some sort of resistance to motion, some sort of shearing, some sort of cell-cell adhesion, something like that. So again, it's sort of very heuristic. And again, if you ask, we haven't got the data to parameterize this. So these are really qualitative models rather than anything else. So the constitutive assumptions, as we've just discussed, I'm going to assume that the live cells are, there's a pressure, which would be as for the live cells exactly as we had it before, but now with a viscous term added in, I'm going to assume that the dead material follows the same thing, but that the viscosity is proportional to the viscosity of the live cells. Theta 1 is kind of how much different. Theta 1 is between 0 and 1, and that's sort of, I guess, the dead cells are somewhere between live and dead, and it's trying to capture that sort of behavior. Notice here that the sigma term, I'm saying that the dead cells can have some influence. They occupy volume. And so rather than just having phi1 in here, there's a weighting on the dead cells with a parameter theta 3. So the theta 1, 2, and 3 are kind of representing the sort of extent to which the dead cells are somewhere between fluid and live cells, right? Because they're degrading from being live cells to dead cells, right? And as we had in the previous models, we assume that the outer boundary, our domain, grows at a rate determined by the live cell velocity on that boundary. So you can go through all of the algebra, it's just a little bit more horrible. So then the question is, what qualitative behaviors does this model exhibit? What do we get for all this extra work? And so for simplicity, I've fixed all of those thetas to be identical, just so that we can vary one parameter at a time. And so if we start at the top here, I think this is theta equals 1, and this is theta equals 0. So theta equals 0 says that the dead material is just like water. Theta equals 1 is saying that the dead material is like cells, so a viscous fluid. And these are the values in between as I vary those properties. And I hope you can see that it does indeed. So this is just the tumour growing, no treatment, nothing. So you can see that the qualitative behaviour is quite different. When the dead material is more fluid-like, it will be able to move more. There's no resistance to movement. And as dead materials is created, it will kind of flow out from the tumour centre, and it will recycle. It will be taken up as material for cell growth. And so it will be, because it's not viscous, it will move and circulate much more readily. And indeed, for this parameter set, you can see that if I view the dead material as an isotropic fluid, then we're getting tumour elimination for this set of parameter values, and we're getting persistence. So quite different qualitative behaviours, right? So now what happens if I start irradiating these tumours? So what we do here is we apply a single dose of radiation at this time point, and then we look inside the tumour box at these different time points. And this is giving you a cross-section through the tumour, showing you how the 3 different phases are distributed along the tumour radius and how that distribution changes over time. So this is the distribution or the composition just prior to radiotherapy. The yellow line is the fluid, and you can see throughout that doesn't change terribly much. The light blue is our dead material, and the dark blue is live cells. So as you might expect, so superimposed on top of this is an externally supplied nutrient that's diffusing from the tumour boundary towards the centre. So cells on the boundary, nutrient rich, they're proliferating a lot, and so we've got predominantly live cells here. Only a very small volume fraction of dead cells, and a sort of maintained almost approximately constant volume of fluid. As we go towards the tumour centre, we have predominantly dead material. This interface here is marking the transition to hypoxia. This interface is marking the transition to necrosis, cell death. So we come along, we irradiate, we preferentially hit these live cells, and we convert them to light blue material. And that's what's happening just here in a sort of fairly obvious sort of a way. And then we watch how the system evolves over time. And you can see that by t equals 50, the tumour has regrown past its initial size and its composition is being restored. So I guess you can notice here that the positions of these internal free boundaries marking the transition from proliferating to hypoxic to necrotic, when just after we've irradiated, because we've got rid of so many of the live cells, this necrotic boundary has disappeared, right? Because nutrient is everywhere above CN, or the threshold at which necrosis onsets, and the position of this interface for hypoxia, marking where cells stop proliferating, that's also moved. Okay? So you can start to see how the tumour composition is changing over time. And then using this, we can then try and expose these tumours to the same course of radiotherapy that we did earlier this morning. And what you can find is that for different parameter sets, we can reproduce all those different growth dynamics. And here are the other two. Okay, so again, it sort of suggests that one possible explanation for these growth dynamics, and it just allows us to see what the spatial composition under these assumptions might look like. Okay. We can also do various other things. We can follow and work out time to regrowth of different sort of subtypes. I guess it's all fairly obvious. I think more interesting things that can happen here is we get what's the consequences of radiotherapy on the post-treatment dynamics. So in this case, we have a tumour. If we left it untreated, then it would follow the dashed line. We start radiotherapy, and then afterwards we see it starts growing. Not so good. So this is sort of suggestive of bistability in our system. Okay? So we make some perturbations, we take it to a new point, and then it evolves to a different configuration, right? To a travelling wave profile. Again, we can use the model to investigate how the properties of the dead material influence the response to radiotherapy. So in here, let me see, we've either got the blue line where we view the dead material as being similar to the cells or similar to the fluid. And you can see how those different constitutive assumptions are impacting both the response to treatment and, well, I guess ultimately the regrowth. These are both regrowing, but you can see how it makes quite a strong impact on the response to treatment. So we saw this model earlier this morning. And as I said, the way that this work actually happened is we developed the three-phase model first, saw it reproduced, and then we thought, once we understood what was happening, that helped us to arrive at this model. And what I'll show you now is how we can actually use these two models together to do some experimental design. So if you think about it, the sort of key assumptions about this is differences in the way in which the tumour is handling its dead material. But obviously we can't test any of those assumptions because we don't have that data from the clinical studies. But we can generate synthetic data, and we can see if we could measure how much dead material there was, how much better would that enable us to fix the parameters in the model, and what would we learn to distinguish between different sort of parameter sets. So yeah, the plot here is just showing you, I should have put this in this morning, but never mind. It's just for this model, as you vary, I think it's zeta, the rate of decay of the necrotic material, and the rate at which dead material is formed just naturally. As we sweep across those two parameters, the different qualitative behaviours that this two-compartment model exhibits, just so that you can see the zoo of behaviours, qualitative behaviours that it exhibits. Now, if we fit that two-compartment model to the data, and we look at our certainty in the tumour composition, because we've only got tumour volumes, unsurprisingly, our certainty in the composition is pretty rubbish. And again, this is very similar to what we did first thing this morning when we were looking at the control and wild type, the wild type and resistant treatment, and we only had tumour volume. So this shouldn't be so surprising to you, right? So I can get a good fit to the volume, but I have no clue what's happening inside the box, right? So in very much the same way as we did with those in vitro experiments, we can now we can fit this model to data that was generated from our 3-phase model. And we can say, if I only have tumour volumes, so up at the top, again, you can see similar to when we fit the model to the clinical data, I can estimate, I can fit reasonably well to the synthetic total volume, but I can't really discriminate what the composition is. It could be anything, right? However, if I supplement my total tumour volume with measurements of how much dead material there is, then my predictions are so much better, right? So that extra information would really enable me to narrow down what was going on, right? And I guess this would be a way we might have an alternative model where we say it's not dead material, we think there's a resistant cell population, or actually even if you found that there wasn't dead material, then that would put this model in the bin, which is where it might be where it should go. I don't know, but whatever. The principle's still the same. So I guess the point here is that we can actually use, now that we've got our complex 3-phase model, we can use that as a source of synthetic data with a known ground truth to kind of do experimental design where we don't have that experimental data, and it sort of makes a case to an experimentalist, if they could do this, then this would be the benefit that you would get from doing that. And I think that's another, so because you might say, what's the point of having a super fancy model that you've not got a hope in hell of being able to parameterize? This at least makes a qualitative case for the value that could be gained from doing that. So I think that's about all I wanted to say for today. So what we've tried to do, I think, is show how existing classical models of avascular tumour growth, our Greenspan and nonlinear diffusion equation, how those can be brought together under a multi-phase framework. We've looked at an extension to 3 phases, which revisits the data that we were looking at earlier this morning, shown that the 3-phase model gives us, I think, additional insight into what's going on inside the box in terms of tumour composition, and how we can combine that 3-phase model with our ODE model in order to do experimental design. So I thought that was probably enough. There's a lot more work that I haven't told you about where we've used this framework to do lots of other things, to develop models of vascular tumour growth, where we can really exploit the mechanical bits to look at things like vessel occlusion. We can look at mechanical sensitivity in the tumour cells, how that might inhibit proliferation. We can bring in immune cells, all sorts of things. I think we've done some other work looking at oncolytic virotherapy. I mean, there's a bunch of stuff. So if you're interested, but the basic idea is the same, and hopefully you get the gist of how the framework works. So I guess, yeah, take-home message from today is you can develop models at lots of different levels of complexity. The challenge is working out what's the right one for the problem that you're using. Okay, I think that's enough for now. Thank you for listening. Happy to take any questions, and I've actually finished on time. Good, thank you. Yes, 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 yes. Yes, so, and I guess in my, in the compartment model, it kind of, all of that is glossed over, really. It's not sophisticated enough to do that, unless that's around. Hadn't really thought about it before, to be quite honest with you, but it's a good question. Yeah. Yep. Yes, so how big is the cell? Yep, and how big would a tumour spheroid be? Probably be, well, if you look at some of the the pictures, so they're thousands of cells. So I guess an avascular tumour spheroid might be 0.5 millimetre, 1 millimetre, right? So there's lots of cells. A continuum model, I think, is justifiable. Again, if you're thinking about a tumour in a patient, by the time that's clinically detectable, it's going to be much bigger than that. I think by the time something's detectable, then you're at a continuum model is legitimate. There are, you are right. I think where I think that becomes really important is if you want to say, I've eradicated a tumour, what is the extinction probability? A continuum model isn't going to do that. So once you get down to very small numbers being probably, I don't know, less than 100, then probably doing cell-based models or stochastic models is the right way to go. So these models won't really become extinct. I guess here you would have to say, once the volume gets down to less than a certain thing, then I call that extinction. But these are deterministic. There's no allowance for stochastic effects. I think interesting is potentially once these models predict down to a small enough size, then you seg into a stochastic model that takes that sort of stochasticity and gives you a possibility, a probability of extinction into consideration. So that's a local assumption. So things can move, and this is where, depending on how I model, say, my dead material, then it's the water that moves. It's the easiest one. Yes, yes, yes. Yeah. It has to flow in because otherwise it wouldn't get bigger. Yeah, yeah, yeah. So I glossed over, I don't think I imposed a single or stated a single boundary condition here. So I guess you've got continuity of stress, etc. You need to think about what the boundary conditions are for your stress tensors, etc., etc. And I didn't specify those, but that would be that the fluid can come in freely, and that's basically what's giving you the mass that allows the cells to grow, because the cells are growing by taking up water. Yep, you can do all of these things. Yes. So we haven't done right back to first principles homogenization, but we have looked at multi-phase models more for things in tissue engineering, looking at, say, cells growing on scaffolds. So where we can kind of model the scaffold as being some sort of poroelastic material. So there are papers doing all of those things, but we haven't done the homogenization. I guess the difficulty here is dealing with the cell growth? Yes, that's possibly a problem. Yes. So when you say benign, what do you mean? Yeah, I haven't looked at symmetry breaking. I've pretty much looked at 1D models, right? You can, and again, there are papers in the literature where we can look at, so typically a lot of the work's looked at 1D sort of radially symmetric or 1D Cartesian geometry just to see the basic principles, but there's been quite a bit of work, not so much recently, where we can look at, say, if you want to do things analytically, you can add symmetry breaking perturbations to the tumour boundary, and you can look at instabilities that emerge, and you can look at, depending on how strongly the cells adhere to each other, so cell-cell adhesion, then either those perturbations, if cell-cell adhesion is not strong, so the cells don't want to stay well stuck together, then you will form instabilities. So there's quite a lot of work that's been done looking at symmetry breaking, and again, solving these equations in 2 and 3D, you can also see that. And I guess there's lots of other ways to do this, phase field models, many other ways of modeling continuum models. This is just one such. But again, good question. Yeah. So I think some of the interesting ways or things that I think would be interesting to do is learning surrogate models. So again, it would be interesting to take the output from my multi-phase models and see what sort of ODE model could you learn from that. I think that's some ways of going. Equally, if you've got a very complex model that you want to solve in 2D or 3D, if you can machine learn a way of exploring parameter space with a surrogate, rather than performing a 6-dimensional, whatever, n-dimensional parameter sweep, then you could use that to march around parameter space, find interesting places, and then go back to your full model. So I think there are a lot of ways that you could use machine learning, interface that with sort of mechanistic models to sort of help. Yeah.
Okay, I think this is a good point to break.
Yep.
Let's thank Helen once again.